Hello everyone, my name is Ashfaq Ahmed and I work in the field of bioinformatics and structural biology. Today we will talk about UniProt database and my focus will be that how can you effectively use a UniProt database. We also call it SwissProt database. Uh, While teaching you this UniProt database, I have classified this session in a kind of few questions. The questions which I normally receive from the students during the classes also on this channel. So therefore, I thought why not to address the knowledge of UniProt database by answering those questions. So most of you will get benefit the most from this video. So let's start. Okay. So first of all, what is a UniProt database? UniProt database, we use it like uniprot.org and you will see here find your protein. So from this term, it means that this is the database primarily for protein sequences. So your question, where to find my protein sequence? The answer is UniProt database. So the next thing is how will you access or how you will search UniProt database. That is very simple. If you know the name of your protein or if you know the name of your gene, because normally the product of gene to what we call protein is the same with the gene name. So, for example, I'm searching EGFR. Now, on this panel, you may receive too many entries like EGFR, 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 but if you look carefully, EGFR, APIME, EGFR mouse, and MACMU, etc., etc., you can scroll it down and you can see. Okay, so EGFR APIME. This will be your protein name and this will describe the scientific name or the common name of an organism. So APIME stands for Apis mellifera, to what we call honeybee. This here you will see the protein name and here you will see the gene name. Most of the time, as I told you, the gene name and the protein names are similar. But sometime, maybe the gene name is different or the protein name is different. Okay, you will see also this name entry. So to this we call it accession number. Remember students or particularly undergraduate students, if you are working in UniProt database, so it is somewhat essential for you to note down this number, this accession number of your query for further or for future uses. So you can quickly access your data or you can quickly reach to your data next time. Okay, if you see this tag, you see this is gold color, gold color, gold color, again gold, but these two data, they are not gold color, they are just gray. Again gold color and then again it's a gray. What does it mean? You should know that. It explains the status of the sequence. If it is in gold color, it means this is a reviewed sequence. If it is in this form, it means this sequence is not reviewed. Probably it may have minor mistakes, and after revision by the concerned staff or experts, uh, that minor mistakes can be or will be removed or fixed. So therefore, I would suggest if you have the option to select between the reviewed and unreviewed, so I would strongly suggest always go for the sequence which is a reviewed one. Okay, now let's say I'm opening this data. So right now, where you will find your sequence, protein sequence, particularly UniProt. Okay, what next you can take from UniProt? So I have mentioned these things in the form of questions. So I will explain these questions one by one. Normally, I receive a question. How will I know that the structure of my sequence is present or not present? It's a valid question. And people will tell you that go and search PDB database. But remember, if you are new in the field, so it is not a straightforward task to search in the PDB because PDB normally gives you a keyword search, and a keyword is actually a tag or a word or a sentence written by the submitter. So maybe if the protein name is, let's say, A1 or A1B1, but if I call it SRC3, then you will search for A1B1. Possibly PDB will say, I don't have the structure for A1B1 because if I'm the depositor and I name it SRC3, because there are a number of proteins which have more than one name or more than one common name. So uh, the reason is because at the time of structure deposition, sometime the author used the name which he or she used inside the lab. For example, when I was a PhD student, so we were working on a protein that was called YYCG. YYCG. So for the 4 years, we were talking in meetings about YYCG this, YYCG this, but its real name or literature or accepted name was WALK, WALK. Now, if I search, I will say YYCG, most of the papers cited YYCG, but in PDB, maybe some sequences will be with the name of the walk or with the name of the YYCG. So I'm not going in a much more detail, but actually I provide this detail to you that in PDB, the search is by keyword and the keyword are generated by the depositor and sometime you may feel that my structure is not present, but in reality, it is present. So, in conclusion, search by a keyword for a structure in PDB is not that simple for a newbie or for a beginner. So then the question comes, how will he or she or you will know that the structure of your sequence is present or not? For example, this is your sequence. This is the accession number. The sequence is reviewed. This is the name and it's from the human entry. Can you see this left side? Here is the panel. So I would suggest you need to click here, structure. Okay. Now wait for some time. If now drag it down. If you see this kind of window that says PDB, 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 etc., etc., and you can scroll it down so many. So you see, without a search, you can see whether the PDB structure, the structure in the PDB is present or not. So for this entry, I think more than 50 structures are present. Now, which one to select? Maybe this is another question for you. So, rather than to study the PDB database one by one, why not you study here? So, normally on two criteria, we select a structure for our research, two or three criteria. Criteria number one, are you selecting X-ray crystallography, NMR, or cryo-EM structure, cryo-EM? So these are the three structure, and most of the time people will go for X-ray crystallography. If you don't know the difference, I would suggest go for X-ray crystallography, because I'm not going to provide you extra detail for these methodologies. If you want to know, let me know. I will make a video for it. Okay, so X-ray crystallography. If most of the time people wants to go for X-ray crystallography, option two for the selection. People wants to go for the higher resolution. But once again, what does it mean in terms of protein structure, the higher resolution? For example, if I have the choice to select between this structure and this structure, both are solved by X-ray crystallography. Which one is the higher resolution? So don't say 2.6. The higher resolution is 2.4 angstrom. The lower the number, the higher is the resolution. That's it. Why you will go for the higher resolution? Because the higher resolution means that the structure is pretty much clear at molecular detail, at molecular level. Okay. So I have explained also the resolution, and the third option for structure selection is which region you are interested in. For example, if your supervisor asks you that, look, this protein has three domains. One is catalytic, one is DNA binding. We are interested in DNA binding domain. We are not interested in catalytic domain. So he or she, your supervisor, clearly mentioned to you that we are interested in a DNA binding domain. So from here, you will select the structure only of DNA binding domain. If you are interested in the full length structure, so how should I know that this is the full length structure or not? So that is very simple. If you come here, sorry, okay, sequence, click here the sequence. It says that the structure is total length is 1210 amino acid for EGFR. The full length structure sequence is 1210. Now click the structure panel again. Now you will see that do I have a complete structure for EGFR? So just read here from 25 to 646. No, it's almost half of the structure from 692 to 1022. It means it starts from the middle and ends on the C terminus, but still almost 200 residues are missing because total is 1212, I guess. Okay, so quickly you can see status of all the structures solved by the EGFR are solved by your protein. In case, okay, you want to open it in PDB, so that's very simple. Right click, sorry, right click here and open in new tab, and directly you can go to the PDB. Next, if you want to open directly, okay, this is the protein data bank which is governed by or which is managed by the European side or EMBL. But that doesn't matter. It has the same data. Also, you can go this way, click here, rcsb.pdb. So it will bring you to the real PDB which I call it the real. By the way, both are same similar thing. Okay, so you can see I came directly here. Now, before coming to here, I have the information that this structure is starts from this amino acid and ends on this amino acid. Okay, so uh, this was the answer for your this question, that how will you know that the structure of my sequence is present or not? If it is not present here, it means that the structure is not present in the PDB. Now you have options to model it. Which method to use? This is another debate. I either to use homology modeling ab initio, excuse me, or to use the current AlphaFold. Okay. Now, what is the second question I received? From where I can take the sequence of my protein. So, my dear, if you are a new person, if you are not experienced or expert in bioinformatics, that is very simple. Just open your sequence, just like I open EGFR human, and I can assure you that the information for your gene, for your protein will be present on the UniProt. So once you open it, just click here, sequence and isoforms, or you will see sequence. That doesn't matter. Isoform is a different thing. It will not be present for every sequence. But if for some sequence there is an isoform, then it will be present. If you don't know the isoform, so I just give you a conceptual idea that when you are studying protein expression, transcription, and translation, or the protein synthesis, so after the transcription, you study a smaller topic that is called protein splicing. So, because of that splicing, when introns are removed, so the exon shuffling take place. For example, if a protein has 5 exons, and after removal of the introns from the middle, when they combine, so sometimes they shuffle. Let's say from 1, 2, 3, 4, 5. This is protein 1, but 1, then exon 5, then 2, 3, 4. This is that makes another protein. So we simply, I will give you a concept this way. So this kind of protein will have 2 isoforms. So click here. Now you will see the sequence information of your protein. Now don't need to copy this way. This is I would say a stupid way. Okay, sorry. Just click here, copy sequence, you will see copied. Now open new like the text file where let's say sequence, or open it and click Control V. Can you see your sequence? Yes. So the point I'm making is that it's very simple to take your primary sequence, and what is the length of your sequence is this one. What is the molecular mass in Dalton? So it is 134277, but normally in literature you will see kDa. So if I ask you what is the molecular weight in kDa for 134.277, I need your answer in comments. If you say that I know it. So I will not tell you what will be the molecular weight for this protein in kDa. Okay, if you are listening me, so I'm also giving you another trick. For example, I give you like your protein is 1210 amino acid long. What will be the molecular weight or expected molecular weight of this protein? Can you answer this question? Okay, let me help you. Uh, Multiply 110 with the length of your protein. You will get the molecular weight in Dalton. And then how you will get in kDa? That is a question I dropped it for you here. Okay. Now, if you want to know, okay, we will cover from here. Okay. Question 3. How will I know that my protein is interacting, that the protein interacting with my proteins are reported? Let's say I asked you that, okay, this is your protein. Now I need a data that to what proteins this protein is interacting, your protein. So how you will take it quickly? Simply, the answer is take it from literature, but how to find the relevant literature directly? If I give you 1 hour or 2 hour or let's say 30 minutes, so the answer is once again UniProt. Just click interaction. If there are any reported interaction studies, it will be here in the interaction panel. But not only the study, you can also find the whole papers. You can see PubMed. So for this interaction that the EGFR is interacting with the CCDC88A, autophosphorylation, whatever, or it is the part of this complex, you can find these two papers. Just how to open this paper, don't worry, right click, open new tab, and it's the abstract. And if you want to see this paper on PubMed, click here. So it will lead you to the PubMed. You can see here. Now from here you can evaluate or investigate or click here for the PMC or open access complete paper. So this way you can, even if I want to know about the EGFR, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, almost 20 to 25 papers are there. So it's better I should start from this paper if I'm studying the interactions of EGFR rather than to search on Google and here and there. So be specific and be smart. Okay. Now, next question. How will I know structural representation of my sequence? Again, I am presenting a scenario for you. For example, I'm your supervisor and I ask you that, okay, this is your sequence and tell me how many domains it has. Tell me, is it has any disorder region? Okay, I ask you this way. Okay, it has 3 domains, but tell me from where the C-terminal domain starts. So I made 3 questions for you, but the answer is one. Again, just click here, family and domains. If there are some reported family and domains here, so you can see that from 688 to 704 is the dimerization or the phosphorylation domain. Now, use your common sense. If you are interested to work with the dimerization and phosphorylation domain, then I would suggest to that you will need structure of this region. So don't make yourself confused. What are you doing with the full length structure? And if it's not present, you are going after it. So most of the time, because I know the structure is present for this domain, how I know? Because a little earlier we saw a structure that was started from 645 and that ends on 1022. So it means it includes this domain. Okay, if you are studying the protein kinase effect or you are inhibiting the protein kinase, so you will need this region because the 7112 from up, up to 979 is the protein kinase domain. And do you know what is kinase? So I have also covered a long ago that there is a there is a trick in these names. For example, whenever you hear a name kinase, remember this protein will functions to phosphorylate others. Now, how it will phosphorylate others? It will hydrolyze ATP to ADP. Then it will use or donate one phosphate to another protein and to what we call phosphorylation, the addition of phosphate. But such proteins which are phosphorylating other proteins, they are called kinases. So, for example, if you hear something, if you are sitting in a room or if for some PhD defense, and if you are undergraduate student, and if the person is telling you that this is a kinase protein or this is a kinase domain of this particular protein, you should know that he actually means that this region or this protein will phosphorylate another protein. If he did not explain, you can ask in the end, sir, you mentioned this is a phosphorylating domain, or sorry, this is a kinase domain. So, can you please tell me what protein it will phosphorylate? Believe me, he will be amazed at so much. A good question, but how you pick this question? Because you knew what the kinase means. The same way, the opposite of kinase is phosphatase. So phosphatase will always remove the phosphate group. So there are so many these names phosphatase, hydrolase, kinase, transferase, isomerase. It's better to learn it. It takes less than 30 minutes. Believe me, you will enjoy biology. Okay. This region is a disorder region from 1097 to 1137. And do you know what are disorder regions? The organization of amino acid present in this region isn't that simple to be folded. And even the current state of the art programs, AF AlphaFold I'm talking and others, most of the time they fail to fold correctly the disorder region. So therefore, if you know these information and if you are using the alpha fold, I would suggest to remove the disorder region from the sequence. Otherwise, your structure in that section will not be a good one. Okay. So I hope I have addressed this question too, that how you will know the information. Okay. Now, sometime you are interested in the subcellular localization or location of your proteins. Click here, names and taxonomy. Click here. If there is some residue which is post-translationally modified, click here for expression. So, and for similar proteins. I hope I have explained and I have covered each and everything or most of the thing in the UniProt database, and I have divided this talk or this tutorial or this lecture in a question form because I wanted that you people that I wanted to address the teaching of UniProt in a kind of problem solving manner. Okay, I hope I will not feel or I will not have such questions again. And if you are liking my videos, so please subscribe and spread this channel to your fellows and colleagues. And thank you. Bye bye.